Okay, today we're going to be looking at special senses. And I'm just going to go through each one of them one by one and talk about how we sense the world around us and turn those into electrical and action potentials. So, first one we're going to look at is smell. So, go ahead and erase that. So, with smell, we have big old nose looking thing here. It's not great, but it's the best I can do. Up here at the top, we have what's called the olfactory epithelium. This is a place where our sensory receptor nerves or receptor cells are. So they'll come down and they'll go through here, and then they'll go up through a plate here up into the olfactory bulb, which will send the action potentials depending on how we sense things coming in. So to look at this a little bit better, that's the overall kind of anatomy of it. To look at one of these cells a little closer up, you have these cells that come down and they have little receptors on them. Okay, and with smell, it's chemoreception. So different chemicals in the air or things, they'll bind to channels, and that will begin this receptor potential and the cascade. So with smell, we have the cell here. Let's draw this a little bigger actually, so that we can see it a little bit better. Come down here, you have these big bulbs and it's coming up. Okay, so down here we'll have this G protein. And some smell, some chemical in the air will come and it'll bind to that site. This in turn will activate this G protein and it will send the subunits to begin a cascade. So this cascade, there is this other protein here. On here, this is adenylate cyclase. And from that, it takes ATP and makes it into CAMP. That in turn, this CAMP will come over to another receptor here in the side. And this is a ligand-gated cation channel. So this CAMP comes in, it binds, which causes a conformational shift in the protein to open it. When this opens, like that, we are able to get calcium and sodium to come into the cell. That in turn will create some sort of depolarization event, beginning a belong down the cell called a receptor potential. That receptor potential will travel to the end where we have our voltage-gated calcium channels And we'll just draw the end of the cell here with vesicles and snare proteins. Okay, calcium comes in, this opens, calcium comes in, binds here, and we release those vesicles to the nerve cell here. Just kind of do that. That's a nerve cell. And it'll trigger an action potential down the nerve. That's how the olfactory system works. We get some sort of chemical binds to a G protein. That G protein activates adenylate cyclase. CAMP then activates this cationic channel, allows calcium and sodium in. BLONG goes up, receptor potential activates the calcium channel, releases a neurotransmitter, and we send an action potential back to the brain to be processed and see what smell we have. And it all is dependent on what kind of chemical binds to that G protein. So that's smell. What about taste? Well, taste, on our tongue, we have a bunch of different types of taste buds. All these different ridges and valleys and stuff. And so I'll just draw a little tongue here. Little tongue here. And up here, we have these little ridges that come in. Kind of like that. Those are giant papillae on the tongue, right? And down in here, we have little pockets that are full of these receptor cells. Those are the taste buds. So, how do the taste buds work? We have 5 different kind of modules of taste that are the different things, I guess, we can determine. We have sweet, sour, bitter, sweet, sour, bitter, umami, and salty. Okay, so these are all that we can really taste, and sour and salty, they go on the same pathway. And umami, bitter, and sweet, they're all from G protein. So we'll put G protein, and these are ionotropic or just channels. Channels, okay. So let's first look at salty and sour. All right, so I'm not going to get too technical with my cells here. I'm just going to kind of draw a box, simplify the cell. So we have a receptor cell here, and coming off of that receptor cell, we have some sort of nerve cell, something to send an action potential. Up here, the top of the cell, we have some sort of ligand-gated cation channel. So this is a ligand-gated cation channel. So, how do we determine between salty and sour? So, let's say we get something salty. It's usually potassium chloride, sodium chloride, some sort of salt, right? So, the salt will come in and it'll bind. So we have KCl, NaCl, they'll come in and they'll bind to that site there. This will open the gate here, allowing sodium and calcium to flow into the cell, producing a receptor potential. That receptor potential will travel through the cell, where down here we again have a voltage gated calcium channel. We'll CA sitting out here. So it comes in, we have our vesicles here full of neurotransmitter. The snare proteins, just draw one, actually I'll draw them all. So the receptor potential comes in, depolarizing event, calcium can then, this then opens, calcium can rush in, bind to the snare proteins, and bring the neurotransmitter out to send an action potential down the neuron here, the taste neuron. But if this, instead of a salt, was hydrogen ions, it would bind, and then hydrogen could also come in to the cell because it is a cation as well, helping to depolarize the cell, signaling the depolarization event. Calcium comes in, and we send an action potential. So this is the example of sour and salty. So our other ones, we have bitter, umami, and sweet. These are metabotropic. They run off of G proteins. So here we have a G protein embedded in the membrane. And we'll start with bitter. A bitter taste is when we have calcium come in and bind to the receptor. So we have a receptor here. Calcium will come in. Let's do that in black. Calcium comes in, it binds. We activate the G protein cascade. Down here in the endoplasmic reticulum, we have high calcium concentration. This will come down here and it'll trigger a calcium channel on the endoplasmic reticulum, allowing calcium to leave. And that will then activate the snare proteins and release those vesicles down into the dendrite to signal an action potential. So if it's not calcium and it's glutamate, GLU, glutamate, GLU, it comes, same thing, activates this. We have a cascade to the endoplasmic reticulum, releases calcium. Calcium binds to the snare proteins, releasing a neurotransmitter. We get an umami taste with glutamate. That's the word I was looking for, glutamate. And if we have carbohydrates, carbs, so sugars, glucose, fructose, lactose, we have some sort of carb come in, it binds, and we taste sweet taste and stuff. It's from carbs. So sour and salty, ionotropic, and they allow protons in, and that will signal the cascade. And then bitter, umami, and sweet, metabotropic, G protein to endoplasmic reticulum, calcium out, signals an action potential. So that is taste. We have 3 more special senses, but we're not really going to talk about touch. So first, I'm going to talk about eyesight, and I'm going to end with the ear. So, eyes. On the eye, we have the eyeball here. Just draw a whole big circle. We'll erase this front part. All right. So you have the eyeball. Okay. So you have the eyeball. Right here we have the cornea, and you have the pupil. And behind that, we have the lens. So the lens here and cornea here, they help to bend light, because the way we see is we have photoreceptors, which I'll get into in a minute, back here on the retina. And we need to focus the images and the light that we see onto the retina. So we have a point out here. We'll have that as a light point. And then these green lines, they'll be light rays. So we need a way to get rays coming up here, straight this way, kind of angled, and really angled down here. We need to get all of those converging on one point so we can clearly see this image. So what happens is this right here, because of the way it's curved, the curvature of it, it'll begin bending light. So this one, it's coming pretty well straight. It'll come almost straight through. This one will begin bending up, and this one will bend up as well. And this one, even though it's bending, it's not going to get in because of where it doesn't fall in the pupil as it's bent. And then, because this one is also a concave, the light's going to come through. It's going to bend again. This is going to bend. And this one's going to bend. And it's all going to come in and focus on that retina. One point here on the retina for this one point out here. So, if that point is farther away, we don't need to bend the light as much because it's coming in more flat. So, in order to bend the light more, we need more of a concave shape. So, I'll erase this, and we'll just look at the lens. So if I draw a giant lens here, it's kind of a football shape with these suspensory ligaments all around it. They come off in all directions, connecting to the ciliary muscle. It's a kind of a sphincter muscle. It's a smooth muscle that runs around the eye like that. So this is the lens, suspensory ligaments, ciliary muscle. So if we're trying to see something far away and we don't need to bend that light as much to get a clear image, we don't need a very round football. We want a thinner football per se. So what we're going to do is this muscle here is going to relax. As it relaxes, it gets wider, because it's circle muscles. It's a circle muscle. It's going to contract. We're going to want to, as it contracts, that lens is going, these ligaments are going to loosen, and that lens is going to get rounder, right? So, as we want to see farther, this is going to loosen up. The ciliary muscle loosens. That tightens these and elongates this and flattens it down. So it would look more like that, you know? So that way, light coming in isn't being bent as much. But if we want to see something that's closer, these muscles are going to tighten, making these ligaments loosen, and this is going to get wider, go out more spherical, kind of. It's going to widen out so that it can bend the light more to focus it back here on the retina. That's how the ciliary muscles and sensory ligaments work. So, I mentioned photoreceptors earlier. We're going to talk about how they work. So, on the back part of the retina, you have these layers of cells. And it's going to sound funny because the part of the eye, the cell that actually senses light, is at the very back of the eye with other cells in front of it. So we have the back portion of the eye here. We'll just draw this little membrane here, okay? And then we have cells that run along it that are called pigmented epithelium. So the pigmented epithelium, it absorbs any extra light that comes into the eye. So if it comes in and it gets past all the cells, past the photoreceptors, it's going to hit this. It's a really dark color. I'll just kind of do this. It's going to absorb that light so that it's not just bouncing around in the eye. But right next to this, we have cells, photoreceptor cells, called rods and cones. Now, they kind of look like this. That's a rod, and cones, they're a little more pointed. They look very similar, it's just cones are a little more pointed there at the end of them. And they cover the entire back of the retina, which is just the back portion of that eye. These are our photoreceptors, they help us see. Other cells that are in this area, we have cells that kind of connect the two. Called horizontal cells. Then we have some cells that come here off of them. And these are our bipolar cells. They're bipolar because the nucleus here, we have pretty much two poles of the cell. So we'll have that on each one of these. And then coming off of those, I'll do it in purple. We have our nerve cells, our optic ganglia. They're called ganglia. And then all the ganglial dendrites, they combine into the optic nerve, and it comes out the back of the eye to go back into, send those potentials back to the brain so we can see. But yeah, so these optic ganglia. So, I'm going to kind of just zoom in on one of the rods, and I'll explain how phototransduction, the seeing light, works through a rod, and a cone is pretty much the same thing. They're very, very similar. And we will walk through how that works. So, we have a rod here. Okay, and then, do it in blue, we have this bipolar cell here, and then in purple, I'll just draw the beginning of a ganglia cell right there. So, on this rod, we have a protein here. It's a G protein. That is called rhodopsin. And rhodopsin, it has a molecule in here that is called 11-cis-retinal. It's a vitamin A derivative, and it looks similar to that. It has a 6-membered ring and a carbon tail with some methyls coming off of it, but in one of the carbons it has a cis formation. So what happens is some light comes in. We have this light come in and it hits. This changes the conformation of 11 cis retinal to all trans retinal. So that changes it from that conformation to, that looks horrible, like that sort of thing. It's all trans now. That no longer fits inside the rhodopsin, so it will come out activating the G protein. This alpha subunit here, let's get green, is called transferrin. It will come over and activate phosphodiesterase. That is another protein here. It is phosphodiesterase. So phosphodiesterase, it goes and it eats up cyclic monophosphates. So what happens is we have a protein here. It's a gated channel. And when GMP, so guanylyl monophosphate, cyclic monophosphate, is sitting on it, so CGMP, is sitting on it, it is open, allowing calcium and sodium into the cell. So they're coming in. So this leaves the cell in the constant depolarized state, which allows for these vesicles down here at the end to be dumping glutamate down into the synapse there. So what happens is phosphodiesterase will come over, do it in black here, will come over and it'll take out that CGMP, closing this. So now that's closed, and we stop getting glutamate into the synapse, less glutamate. So I'm just going to put down arrow GLU, less glutamate. Now that sounds interesting because usually if we want to continue some sort of potential, we want to increase the neurotransmitter in the synapse. But here's the thing. Over here on this bipolar cell, we have another G protein. This G protein gets activated by glutamate. So, again, why don't we want more glutamate here? Because we have a channel here. It's a gated channel. And that gated channel is deactivated by the beta and gamma subunits of this G protein. So when those beta and gamma subunits are sitting here, it's closed. But if we were to remove those, we can open this cationic channel and allow calcium and sodium to come in, creating a receptor potential. So the less stimulated this G protein is, the less this is closed. So with this open more, we're able to bring in more calcium, more sodium. That creates this BLONG, till we come down here at the end. We have our calcium gated channels. It opens, calcium rushes in. We have vesicles full of neurotransmitter just waiting to get out with snare proteins. And so that's where less glutamate equals more receptor potentials down this horizontal, this bipolar cell. And then we're able to send more neurotransmitter to the ganglia and send that action potential. So just to go through it all again, light comes in, 11 cis-retinal to all trans-retinal. It falls out of the G protein. And that's also called bleaching, because once it's able to return to its original conformation, it comes back up in and it deactivates this, allowing for a reset of this system. But transferrin, alpha protein, alpha subunit, comes over, activates phosphodiesterase. Phosphodiesterase then goes and it will eat up and break down CGMP, which was keeping this channel open, allowing for a depolarized state of the cell. So now the cell is becoming hyperpolarized, not allowing these vesicles to come out through the calcium channels and calcium stuff here. If we don't have those, if they're closed, we're not letting glutamate out. We're not letting glutamate out. We're not activating. This is called mGlu-R6. Sorry, I forgot to write that down. This G protein here is called mGlu-R6. So if we're not activating mGlu-R6, this isn't being deactivated. If it's not being deactivated, we're letting calcium in, sodium in, allowing for that BWOG, that receptor potential. Receptor potential depolarizes, calcium come in, release the neurotransmitter, send an action potential. So cones are pretty much the same thing. They have, I think, some different stuff here, but it works the same way. That's how we see in general. But we also need to be able to see edges and details. So how do we do that? It's by this idea of receptive fields. Receptive fields is when we have, and to save time, I'm just gonna draw boxes instead of the squiggles of the cells. We have rods and cones set up in these fields. So they have a center of the field and a surround of the field, okay? Below the center, we have photoreceptors. Below the surround, photoreceptors. So I'm gonna draw a simple one with just one center and two surround. So we have a surround here, a center here, and another surround here, okay? That's our receptive field. So this would be the circle, and then this other circle. That's what it would look like. So this is where those horizontal cells I mentioned earlier come in. So we have horizontal cells. They sit here between all of these cells, the center and the surround cells. And then they all synapse to a single bipolar cell. So we have one bipolar cell here. And then off of that, we have our dendrite. So how does this work? Why is this so important? So these determine the frequency of action potential sent from our eyes back to our brain so that we can see edges and things. The more activated it is, the more action potentials. The less activated it is, the less action potentials. So I'm going to erase this here. I'm going to start with this idea of on center, off surround. So on center, off surround. So that means that light coming in stimulates this center photoreceptor. And light coming in deactivates these the surround photoreceptors. So they all work as I just explained. So what we're trying to do to get the most frequency of action potentials down this neuron here is we're trying to get the least amount of glutamate here as we can. So that means that we're trying to activate this cell as much as we can. These horizontal cells, they act as neuromodulators. So they can help determine how modulated, how well this one is dumping glutamate or not dumping glutamate into that synapse. So here's an example. We have light come into this cell. And let's say that the light hits the center, but no light hits the surrounds, okay? We're gonna do that cascade. We're gonna do retinal, cisretinal to transretinal to transferrin, phosphodiesterase, CGMP's gone, we're closing channels, we're opening channels, all the way down to the point where we release a little bit of glutamate. Okay, so what happens on these two cells? No light comes in, which means we're dumping a lot of glutamate because we're leaving those channels open. There's a lot of glutamate coming down into this synapse. And same over here. Lots and lots and lots of glutamate. Well, these horizontal cells, they have AMPA right here. Boom, boom. That is AMPA. And it's here as well. AMPA as well. So, as you know, AMPA is an ionotropic glutamate receptive cell, or an ionotropic cationic channel that is gated, ligand gated for glutamate. So glutamate comes in, it binds. We can get sodium or calcium or any positive charges to come in and begin a potential. Potential comes through, and here's where it gets interesting. So we have vesicles here. They're full of GABA. So they're full of GABA. So GABA is an inhibitory neurotransmitter. So if these aren't being stimulated, lots of glutamate, AMPA gets activated, positive charges come in, we're dumping GABA into this neuromodulator synapse area and allowing for even less glutamate in that synapse. Less glutamate means this horizontal cell that I mentioned earlier is activated more. We're sending more potentials. So in this case, if we're sending, we're trying to send 10 action potentials, this is perfect. We're going to send all 10. So the other way to look at it is roughly 60% of action potentials of the frequency are sent by the center and 40 by the surround, so 20 and 20. So if we're getting 60 here, 20 here, and 20 here, we're going to get that whole 100. But here's the other thing. If we get light that's coming in and it's also hitting that one, that means because of that cascade, we have less glutamate here. Less glutamate means less AMPA stimulation, which means less positive charges. Less positive charges going through that cell means less GABA, because we're not going to open as many calcium channels. Less GABA means less neuromodulation to the center, which means more glutamate there. More glutamate means less stimulation of the bipolar cell, less stimulation of the bipolar cell, less action potential. So we are actually going to cut out that 20, and we're only going to get 80% or 8 out of the 10 frequency of the signal going. And it's the same if we were to hit this one with light. We'd get less glutamate, less activation, less GABA, even more here. So that would be less activation of the bipolar cell, less action potentials, same thing. So this is for light. So if we have light coming in, this is a great way to do it. It helps us see edge. edges. If we have light being reflected and the edge of the light is here, we can see that edge pretty clearly. Or I guess the edge of the light is here, because if we move a little bit, we're getting 60%, 80%, 20%, because now it's dark here, so we're not getting that 60%. You kind of see what I mean? So what we're doing here is that's how we're able to see edges, is the frequency as the light hits these receptive fields. If the frequency goes really high then really low, there's an edge there. We need to know that there's an edge there. This is great for when there's light coming into our eye. You know, during daytime, this is a great way to do it. But we need to see at night as well. So how do we do that? Well, we do that by switching the center and the surround. We have off center and on surround. Oh, and something I should mention here is on center, off surround, what I just had. The channel, the way we go about this bipolar cell is that mGlu-R6, that metabotropic glutamate receptor, mGlu-R6. But here's the difference. Here's where it changes. When we go to off-center on surround, we change this mGlu-R6 here that was on this bipolar cell, and we change it. It is now AMPA. So now we have AMPA there and AMPA here and AMPA there. Everything is AMPA here. So how does this change things? We have light come in here, okay? It hits. And that equals, light equals less glutamate. Less glutamate down here, as we know, AMPA binds glutamate and then opens. So it needs glutamate to open in order to send that potential, that receptor potential. If it's not opening as much, the frequency of potentials goes down. So light here, less of that glutamate is binding, which means this channel is opening less, sending less action potentials, or I guess receptor potentials. So that means that light now shuts off the center. This is important for nighttime vision, because if it's dark, we need to be able to see edges. The best way to do that is to get these middle ones. So if we have something that sees dark and then lighter on the edges, that's a great way to see it. So we're trying to see light things against the dark backdrop. Great way to do it. So it's the same thing here. These would actually be activating there with light, because light comes in, we get less glutamate. Less glutamate here means less stimulation of AMPA. Less stimulation of AMPA means less GABA here. But since GABA is inhibiting and neuromodulating the center, less GABA means more glutamate. More glutamate, more activation of AMPA here, more action potentials coming through that ganglia cell. So that's how the center and surround works. It's all about the cascade leading to how much glutamate is in this synapse and the neuromodulation between the horizontal cells and that synapse. Okay, so that is the eye. So the fourth and final one we're talking about is hearing. It's the ear. So I'm not going to bother drawing an ear because I'm really bad at that whole drawing thing. But we have an ear here, and then this is the auditory canal. So it comes in, boom. So this is the auditory canal. That's a really long one. I'm going to shorten it because I need to draw more over here. So down the auditory canal, we come to a membrane here. This is the tympanic membrane. And the tympanic membrane it takes those pressure waves, or these sound waves that are just pressure waves, and with it, it vibrates back and forth. That's important because right on this tympanic membrane, we have some bones. We have bones that kind of sit here together. They're called the arcals. So we have the malleus, the incus, and the stapes, or hammer, anvil, stirrup, if you will. So why is that so important? Because these are what help us transfer these pressure waves into the waves that the cells in our ears pick up. Because the stapes, it sits on the oval window of the cochlea. So the cochlea, it comes out and it kind of rotates around, right? There's that oval window. And we have a little membrane here. It comes around. And another thing that comes around. So it kind of looks like this big old kind of swirl right here, right? So the oval window, it comes and it hits. We have fluid in here. And as it vibrates that oval window, as it pushes and pressures up against it, it creates waves in that fluid. So in the fluid of the ear, we have the different cells. But so we aren't really focused on the waves, we're focused on how they change things. So this basilar membrane, it's a membrane that sits right down here, is where your hair cells are. So we're going to look at these different channels, that basilar membrane, and how it all works. Let me just... Okay. All right, so we're going to take a little cross section of the cochlea here. I'm going to kind of draw it out. We're going to take a look at it. We're going to explain how it works. So we have kind of a, it's, wow, that is terrible. Run this marker out. So it's kind of circular, kind of not, sort of thing, okay? And we have different kind of chambers in here. We have our different hair cells and stuff. And we have some cells right there. Okay, so this is the ear, or I guess the inside of the cochlea. So what we have here is this top part is called the scala vestibuli. Scala vestibuli. I'll put that in blue. Scala vestibuli. This is the scala media. And this is the scala tympani. All right. So, why is this important? The scala vestibuli and the scala tympani, they have a different sort of fluid in them. They have a fluid that more closely resembles ECF. So it's called the endolymph. So the endolymph sits in here and here. And then that's the perilymph, sorry. Perilymph sits here and here. The endolymph sits here in the scala medium. And it resembles more of the ICF, the intracellular fluid. So what happens? We have this little membrane down here that I mentioned before. right there, that is the basilar membrane. And it's very fascinating because as you're closer to the outer ear, it's thinner and it kind of gets thicker as it goes back towards, and it has all these little cells in it, and each of these cells, they vibrate at different frequencies. And that comes into effect right here. So we have this basilar membrane, and this pressure wave, now it's a fluid wave through this perilymph fluid, is coming through. Oh, and I forgot. This is called the tectonal plate. It'll come in here in a second. Wow, tectonal, there it is. So the fluid wave is coming through and it's vibrating this basilar membrane. When it gets to a point where the pitch or the frequency of that vibration matches these cells, it will start moving these hair cells up and down because it's vibrating now. And as it moves them up and down, these hair tips, they impact that tectonal membrane and they kind of bend. And this will all become important here in a second when we look at the way that these help us hear, the sounds they help us hear. So in this area right here is called the stria vascularis. So the stria vascularis, it has layers of cells. So I'm just going to kind of draw it here, kind of blown up. Let's make it a little bit wider than that. And we'll just draw couple different layers of cells here. Okay, so what the stria vascularis does is it takes, I'm going to draw some blue blood vessels here because I don't have red. Let's just say this right here is a blood vessel. What this is going to do is this is going to help create the potassium for the endolymph. It's going to bring the potassium into the endolymph because it's right here on the scala media. So what it does is it has sodium potassium pumps on here. So we have our sodium potassium pumps, so NAK, ATPases. And then it also has some co-transporters on here, on this membrane, that bring in sodium, potassium, and chloride. So they're all going into the cell. It's co-transporter, it's a synporter. They're using sodium to bring potassium and chloride in, and it also has some chloride leak channels where chloride can come out of the cell and in. But where this is special is on the very inside of these cells, you have potassium leak channels. So potassium can come and get into this space at a higher rate than everything else because it has leak channels. And then this next set of cells, it's set up the same way. It has all the same channels through here, including these potassium leak channels. So what it's able to do is it's able to pump potassium in and then let it flow out. Pump potassium in, flow out. Pump it in, flow out. In, out, in, out. So it's sending potassium into the endolymph so that there's a large concentration of potassium in the endolymph. So why does that matter? Well, because this is a special situation where potassium is actually going to be doing the depolarization of the cells to begin a receptor potential. So I'm going to zoom in on these hair cells. We're going to talk about it, and then that should be it. So let me erase all this, all my beautiful drawings here. I'm going to draw this bottom portion of the tectonal plate. is it comes down right there. And then we have the basilar membrane here. So this is the basilar membrane, and we're going to have a hair cell. So what happens is we have these are mechanically gated potassium channels. So what happens is basilar membrane begins to vibrate with the correct frequency, and it's pushing. It pushes this hair cell up and into that tectonal plate, bending the hair cell. So if it bends the hair cell, it's going to pull on this and open this gate, mechanically open that gate. So what happens? As I mentioned, this endolymph has a high potassium content, but here's the problem. Inside the cell, it also has a high potassium content, but because of that scala tympani, and it creates a tissue potential, it makes this in here really, really negative. So, or really, really positive, sorry. Makes it really positive. So, in order to compensate, we see a lot of negative charges around here. Kind of see it? I mean, there's a lot of negative charges in there and in here. We see negative potential in both of those areas. So, potassium, even though there's a high concentration of it in here, it allows this potassium to now enter the cell through these channels. As they bend, potassium can come in and enter there. So we have this flow due to the different electrical potentials, not really concentration gradients. It's all based off of electrical pulling that positive charge in. So we pull that positive charge in, and there's these little proteins. I'll draw them in green. All my proteins will be green. So you have little proteins here along the edge of these hair cells. And it's important to note that these are the inner hair cells. So we have, as we saw in that drawing, we had 3 inner rows and 1 outer row. Or 3 outer, 1 inner. So these cells, they have these Preston. They're proteins, and they kind of spring a little bit. So this cell, its potential is usually sitting around negative 40 millivolts, around that. So these Preston at negative 40, they're elongated. They're chilling at negative 40. But as potassium comes in, we get a depolarization event. Potassium comes in, it depolarizes. When it hits about negative 20 millivolts, these Preston, they shorten. And that moves this hair cell and the basilar membranes more, and it moves stuff. So as we're depolarizing and repolarizing, these Prestons are moving, the hair cell's moving, the basilar membrane is moving. Well, we still have all of this fluid out here, the endolymph fluid. So over here, draw it in black, we have one of our outer hair cells. And it also has these sodium channels, the potassium channels, that are going to be mechanically gated like that. And these also have vesicles, but the thing about it is these inner hair cells, they don't really help as much with hearing as this outer row does, the single row. So it has vesicles in here, and it works similarly. As this fluid rushes around and moves around, it's going to move these hair cells. These hair cells move, opening these channels. So these are opening, and potassium flows in. to the cell, depolarizing it. As it depolarizes, we have down here some calcium gated, voltage gated channels. They open due to the depolarization event. Calcium can flow in and activate the snare protein. So this has it too. We have these little calcium channels. Calcium can flow in, activate the snare proteins, and underneath each of these we have neurons. So it goes on and it activates those neurons. And in here we also have our potassium leak channels. We have some that come down in here. So we have some K leak channels, and we have some here as well on these, so that potassium can get out and help to, again, depolarize the cell. Yeah. So vibrations here activate this. As that depolarizes, the Preston compresses and helps move things around, and that creates another pressure wave of fluid, another fluid wave that comes through, and it pushes on these hair cells, opening them. Potassium runs in, depolarizes, activates this, goes in, releases neurotransmitter, and we get action potentials that we